Welcome to the B.Pharm special program where we guide you to study better and in smart way. This is Swapnil Patil going to take one of the most interesting and learning topic of B.Pharm third semester on the important unit of identification of microbes, which is interesting unit of subject pharmaceutical microbiology. Are you ready? Module 1, that is stain and types of staining techniques. Module 2, electron microscopy. Module 3, nutrition, cultivation, isolation and identification of bacteria, actinomycetes, fungi, viruses. Let's start with Module 1, stain and types of staining techniques. A stain is a substance that adheres to the cell and giving the cell color. The presence of color gives significant contrast, so they are much more visible. Different stains have the different affinities for the different microorganisms or different part of organisms. They are used to differentiate different types of organisms or view specific parts of organism staining. What is meant by staining? Staining is an auxiliary technique used in microscopy to enhance the contrast in the microscopic image. Stain and dyes are frequently used in biology and medicine to highlight structure in biological tissues for viewing, often with the aid of different microscopes. Staining and their types. Color compound is used to develop a contrast between specimen and background. A process of imparting color to the cell is known as staining. Cells are stained to reveal the size and shape of microorganisms. The most basic reason that cells are stained is to enhance visualization of cell or certain cellular components under the microscope. Cells may also stain to highlight metabolic processes, to differentiate between live and dead cells in the sample. Cells may also enumerated by staining cells to determine biomass in an environment of interest. Cells are stained to demonstrate the presence of internal and external structures. Cells are stained to distinguish between the different types of organisms. These are all the reasons why the staining is very essential. Now let's begin with staining methods used in microbiology. First, staining is a simple stain. Second, negative stain. Third, differential stain, which is divided into the A, that is a gram stain, B, that is acid fast stain. Let's start with simple stain. They are also referred as monochrome stain since only one dye is employed for the coloration of bacterial smear. The act of taking bacteria taken from lesion or area, spreading them on a slide and staining them for the microscopic examination. The surface of bacteria cell has an overall acidic characteristics because of large amount of carboxyl group located on the cell surface. Due to the acidic amino acids, therefore, when ionization of carboxyl group takes place, it imparts negative charge on the cell surface. As per the following equation, COH giving CO minus plus H plus ion, and H plus is removed and the surface of the bacteria becomes negatively charged, and a positively charged dye like methylene blue attaches to the negatively surface and gives it a colored appearance. Methylene blue chloride giving methylene blue plus Cl. Second, that is the negative stains. Negative staining is the technique used mainly in electron microscopy, by which bacterial cells are not stained but are made visible against the dark background. The acidic dyes like eosine, nigrosine are employed for this method. Though this staining technique is not very popular, it has an advantage over the direct or positive staining methods for study of morphology of cells, and this is because of the fact that the cells do not receive vigorous physical or chemical treatments. The coloring power of acidic dye, example eosine in sodium eosinate, is having negative charge. Therefore, it does not combine with the negatively charged bacterial cell surface. On the other hand, it forms a deposit around the cells, resulting into the appearance of bacterial cell colorless against the dark background. Some suitable negative stains includes ammonium molybdenate, uranium acetate, uranyl formate, phosphotungstic acid, osmium peroxide, and oroglucothionate. These have been chosen because they scatter the electrons as well, also absorb the biological matter well, and the method is used to view viruses, bacteria, bacterial flagella, biological membrane structures, and proteins or protein aggregates, which all have a low electron scattering power. Third stain, that is the differential stains. Staining procedure which differentiates or distinguishes between types of bacteria is termed as differential staining technique. Methods for simple staining imparts same color to all bacteria and other biological material, may be slight variation in the shade. On the other hand, differential staining methods imparts distinctive color only to certain types of bacteria. The basic principle underlying this differentiation is due to a different chemical and physical properties of cell, and as a result they react differently with staining reagent. Procedure utilizes more than one stain. In some techniques, the stains were applied separately, while in other as a combination. There are two important differential stains, namely gram stain and acid fast stain. Let's start with gram stain. Gram stain is one of the most important and widely used differential stain. It has great taxonomical significance and is often the first step in the identification of unknown prokaryotic organism. This technique divides bacteria into two groups, that is gram positive and gram negative. Gram positive, those which retain primary dye like crystal violet and appear deep violet in color, while in gram negative, which lose the primary dye on application of decolorizer and take the color of counter stain like safranin or basic fuchsin. The gram positive bacteria have thick mesh like cell wall made of peptidoglycan, which is 50 to 90% of cell wall, which stains purple, while gram negative bacteria have thinner layer, that is 10% of cell wall, which stains pink. The gram negative bacteria also have an additional outer membrane which contains lipids. There are four basic steps of the gram stain, which includes applying a primary stain, that is crystal violet, to a heat fixed smear of bacterial culture, followed by addition of trapping agent, that is grams iodine, rapid decolorization with alcohol or acetone, and counter staining with safranin. Basic fuchsin is sometimes substituted for safranin since it will be more intensely stain anaerobic bacteria, but it is a much less commonly employed as a counter stain. Crystal violet dissociates in aqueous solution into crystal violet plus and chloride minus ions, and these ions penetrates through the cell wall and cell membrane of both gram positive and gram negative cells. The crystal violet plus ions interacts with the negatively charged components of the bacteria cell and stains the cell purple. Iodine, that is I minus or I3 minus, interacts with Cv plus crystal violet plus and forms large complexes of crystal violet and iodine, that is crystal violet iodine complex, within the inner and outer layers of the cell. Iodine is often referred to as a mordant, but it is a trapping agent that prevents the removal of Cv and I complex and therefore color the cell. When a decolorizer such as alcohol, acetone is added, it interacts with the lipid of the cell membrane. A gram-negative cell will lose its outer membrane and the lipopolysaccharide layer is left exposed. The Cv and I complexes are washed from the gram-negative cell along with outer membrane. In contrast, a gram-positive cell becomes a dehydrated from an ethanol treatment. The large Cv and I complexes become trapped within the gram-positive cell due to multi-layered nature of its peptidoglycan. After the decolorization, the gram-positive cell remains a purple and gram-negative cell loses its purple color. In counterstaining, which is usually a positively charged sapronine or basic fission, is applied last to give decolorized gram-negative bacteria a pink or red color. Now let's move towards acid-fast stains. Acid-fast staining is another widely used differential staining procedure in bacteriology. This stain was developed by Foyle Eldrick in 1882 during his work on etiology of tuberculosis. Some of the bacteria resist decolorization by both acid and alcohol. Hence, they are referred as acid-fast organisms. Acid alcohol is very intensive decolorizer. This staining technique divides a bacteria into two groups, acid-fast stain and non-acid-fast stain. This procedure is extensively used in the diagnosis of tuberculosis and leprosy. Acid fastness properly in certain mycobacteria and some species of nocardia is correlated with the high lipid content. Due to the high lipid content of the cell wall, in some cases 60% weight by weight, acid fast cells have relatively low permeability to the dye and hence it is difficult to stain them. For the staining of these bacteria, penetration of primary dye is facilitated with the use of 5% aqueous phenol, which acts as a chemical intensifier. In addition, heat is also applied, which acts as a physical intensifier. Once the cells are stained, it is difficult to decolorize them. Next method, that is the Zille-Nelson method. Prepare a smear, fix it by gentle heat, flood the smear with carbofuscin, that is S19, and heat the slide from below till steam rise for 5 minutes. Do not boil and ensure that stain does not dry out. Allow the slide to cool for 5 minutes to prevent the breakage of slide in the subsequent prevent step. Wash well with water. Decolorize the smear till a red color no longer comes out in 20% sulfuric acid. Wash with water. Counterstain with 1% aqueous solution of malachite green or luffular methylene blue S18 grade for 15 to 20 seconds. Wash, blot, dry and examine under the oil immersion objective. Next technique, that is endospore staining technique. Bacterial endospore are metabologically inactive, highly resistant structures produced by some bacteria as a defensive strategy against unfavorable environmental conditions. The bacteria can remain in this suspended state until the conditions become favorable and they can germinate and return to their vegetative state. The primary stain applied is malachite green, which stains both vegetative cells and endospores, is applied to help the primary stain penetrate the endospore. The cells are then decolorized with water, which removes the malachite green from the vegetative cell but not the endospore. Saponin is then applied to counterstain any cells which have been decolorized. At the end of the staining process, vegetative cells will be pink and endospores will be dark green. This is all about staining and staining techniques. Now let's move towards module 2, that is electron microscopy. For centuries, the light microscope has been the most important instrument for the studying microorganisms. However, even the very best light microscopes have a resolution limit of about 0.2 micrometer, which greatly compromises their usefulness for the detailed studies of many microorganisms. Viruses, for example, are too small to seen with the light microscope. Prokaryotes can be observed, but because they are usually only 1 micrometer to 2 micrometer in diameter, just their general shape and major morphological features are visible. The detailed structure of larger microorganisms also cannot be effectively studied by light microscopy, and these limitations arises from nature of visible light waves, not from any inadequacy of light microscope itself. Electron microscopes have much greater resolution. They have transformed microbiology and added immeasurably to our knowledge. Different types of electron microscopies are there. First type, the transmission electron microscope, and second type, that is a scanning electron microscope. This is all about electron microscope. Now let's move towards module 3, that is very important in identification of microbes chapter, that is nutrition, cultivation, isolation and identification of bacteria, actinomycetes, fungi, viruses. For the bacteria, nutrient media, the nutrients are the substances used in biosynthesis and energy release and therefore are required for microbial growth. Different common requirements are useful for the growth of microorganisms. These are two types. A, that is the micronutrients. In macronutrients, carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, potassium, calcium, magnesium and iron, etc. These are the macronutrients. While second category, which is essential, that is micronutrients or trace elements. Manganese, zinc, cobalt, molybdenum, nickel, copper, etc. are needed by most cells. Media have classified in many ways, that is 1. Solid media, liquid media, semi-solid media. Second, that is simple media, complex media, synthetic or defined media, semi-defined media, special media. These special media are subdivided into enriched media, enrichment media, selective media, indication or differential media, sugar media, transport media. Third, that is aerobic media or anaerobic media. Let's see details of one by one types of media. First, simple media or basal media. It consists of peptone, meat extract, sodium chloride, water, nutrient agar is made by adding 2% agar to the nutrient broth. It is the simplest and most common medium is routine diagnostic laboratories. Second, that is a complex media. These have added ingredients for special purposes or for bringing out certain characteristics or providing special nutrients requirement for growth of bacterium under the study. Third, that is synthetic media. These media are prepared from pure chemical substances and the extract compositions of the medium is known. These are used for various special studies such as metabolic requirement. Example, simple peptone water. Fourth, that is enriched type of media. Substances such as blood, serum, egg are added to the basal medium and these media are used to grow bacteria which are more exacting in their nutritional needs. Example, blood agar. Fifth, that is the selective media. The inhibiting substance water added to the solid medium enables a greater number of requirement bacterium to form colonies than other bacteria. Example, disoxycholate citrate medium. Sixth media, that is indicator media. These media contains an indicator which changes the colors when bacterium grows in them. Example, incorporation of sulfite in Wilson and Blair medium. Salmonella typhi have a black metallic shear. Seventh, that is sugar media. The term sugar in the microbiology denotes any fermentable substance they may be. That is monosaccharides, disaccharides, alcohol, polysaccharides, trisaccharides or glucosidases, non-carbohydrate type of substances. Example, for organisms which are exacting in their growth requirement. That is pneumococci, his serum sugars are required. They contain 3% of serum. Next is the 8 transport media. In case of delicate organisms which may not survive the time taken for transporting the specimen to the laboratory or may be even grown by non-pathogens, special media are devised and these are termed as transport media. Example, Stott's medium, a non-nutrient soft agar gel containing a reducing agent to prevent the oxidation and charcoal to neutralize certain bacterial inhibitions like gonococci and buffered glycerol saline for enteric bacilli. Ninth, that is the anaerobic bacteria. These media are used to grow anaerobic organisms. Example, Robertson's cooked meat medium, and the cooked meat gives nutrition and these meats kept in a test tube and then paraffin oil is put to prevent oxidation and then the cork is put isolation of bacteria. The importance of this step is to isolate a pure colonies of bacteria. The strip plate is qualitative isolation method. Quadrant sticking is mostly done to obtain the pure colonies. The incubation of the culture is made on the agar surfaces by back and forth sticking with the inoculation loop over the solid agar surface and this will make a dilution gradient across the agar plate. Upon the incubation, individual colonies will arise from the biomass. The characteristic features of the colonies on the solid agar media are then noted and this includes shape, that is circular, irregular or rhizode type, size, that is small, medium, large or in millimeters, while elevation, that is elevated, convex, concave, umbonate, umbilicate, surface, that is smooth, wavy, rough, granular, papillate or glistening type, edges, that is entire, undulate, crenated, fimbriate, curled, color, that is yellow, green, etc. Note the color of the colony. Structure, opaque, translucent or transparent. Degree of growth, that is scanty, moderate or profuse. Nature, that is a discrete or confluent or filiform, spreading or rhizode type. Now let's move towards second, that is actinomycetes. Actinomycetes is a genus of actinobacteria class of bacteria. They are all gram positive. Actinomycetes species are facultatively anaerobic and they grow best under the anaerobic conditions. Actinomycete species may form endospores, and while individual bacteria are rod-shaped, actinomycete colonies form a fungus-like branch networks of hyphae. The aspect of these colonies initially led to the incorrect assumption that the organism was a fungus and to the name actinomyces, that is a ray fungus. Nutritional requirement for actinomycetes. Actinomycetes is a heterotroph, which means it can not produce its own food for the energy. Instead, it draws its energy from its surrounding. As a decomposer, its energy comes from dead or decomposing animal matter, which it breaks down. The complex materials within the animal's dead body became useful to other organisms when actinomycetes breaks them down into the simpler materials. This bacteria is also a nutrifying bacteria, which means it converts the nitrogen in atmosphere into the nitrogen in the form of ammonia, usable by actinomycetes and other organisms. Actinomycetes' nutritional requirement and habitat are interrelated, and as a saprophyte, actinomycetes prefers a moist moderate to tropical environment in which a decomposing material such as decaying logs, forest floor litter and dead animals are abundant. Cultivation of actinomycetes. Actinomycetes require a nutritionally rich media, preferably containing a serum or blood. No growth results are observed on savarant agar. Development of microscopic colonies may require several days at 37 degree. Interspecies variation of colonial morphology is observed. Isolation. Actinomycetes isolation agar M490. Actinomycetes isolation agar is used for isolation and propagation of actinomycetes from soil and water. Composition. Ingredients in grams per liter, that is sodium caseinate 2 grams per liter, L-aspargine, that is 0.1, sodium propionate, that is 4, dipotassium phosphate 0.5, magnesium sulfate 0.1, ferrous sulfate 0.001 and agar 15. Final pH at 25 degrees Celsius 8.1 plus minus 0.2. Principle. Actinomycetes isolation agar contains a sodium caseinate as a nitrogen source. Aspargine, in addition to being an amino acid, is also a source of nitrogen. A sodium propionate is used as a substrate in anaerobic fermentation. Dipotassium phosphate provides the buffering system, while sulfates serve as a source of sulfur and metallic ions. Glycerol serve as an additional source of carbon. Identification of actinomycetes. If gram staining morphology is suggesting a possibility of actinomycetes colony, an acid-fast staining method should be preferred to eradicate rapidly growing mycobacteria, followed by modified acid-fast stain. Fungi. Nutritional requirement for the fungi. Most fungi are growth cultured on media containing natural components. Example: potato dextrose agar, malt extract agar, cornmeal agar. Fungal growth media tends to rich in carbohydrates, slightly acidic pH, that is pH 5 to 6. Fungi can grow on minimum media as well as, example, salts and glucose, depending upon their specific requirement, which may include one or more vitamins, example thiamine, biotin. Organic nitrogen. Some fungi cannot be used nitrate or ammonium as a nitrogen source. Cultivation. Brain heart infusion agar. It is a non-selective fungal culture medium that permits the growth of virtually all clinically relevant fungi. It is used for primary recovery of saprophytic and dimorphic fungi. Second, inhibitory mold agar. that is primary recovery of dimorphic pathogenic fungi, saprophytic fungi and dermophytes will not be recovered. Third, that is a potato dextrose agar. It is a relatively rich medium for growing a wide range of fungi. Sabrant dextrose agar is the most useful as a medium for the subculture of fungi recovered on enriched medium to enhance the typical sporulation and provide the more characteristic colony morphology. Isolation. A, that is a hyper tripping. To separate and isolate one species of fungus from the undesired contaminants. This is the method. Second, that is B, single spore isolation. Remove individual spores from the species of interest, and this procedure can only be used if the fungus sporulates in culture. Identification of fungi and isolation of fungi. This is the importance of this method. Viruses. A virus is a small infectious agent that replicates only inside the living cells of other organisms, while not inside in infected cells. Or in the process of infecting cells, viruses exist in the form of independent particles, and these viral particles, also known as virions, consist of 2 or 3 parts: a genetic material made from either DNA or RNA, long molecules that carry a genetic information, and a protein coat called capsid which surrounds and protects the genetic material, and in some cases an envelope of lipid that surrounds the protein coat when they are outside the cell. Nutrition requirement for the viruses. Viruses must be grown in a living cells. They can't be grown in a culture media or on agar plates alone. They must have a living cells to support their replication. Cultivation of viruses. 1. That is animal inoculation. Second, inoculation into the embryonated egg. Third, that is a cell culture. Isolation and identification of viruses is done by the methods like serological methods, in which Western blotting is the technique used for the isolation. While second method, that is the cytoplasmic effects, a diagnostic inclusion bodies are associated with the rabies viruses, measles viruses, vaccinia viruses, smallpox viruses, herpes viruses and adenoviruses. Molecular methods includes PCR, RF, LPS. PCR was used to identify the West Nile virus, that is polymerase chain reaction. The SARS associated with the coronavirus type of virus identification. Conclusion. With all these information, here we came to the conclusion and end of the topic identification of microbes, in which we deal staining. Staining is an auxiliary technique used in a microscopy to enhance contrast in the microscope image. Types of stains are simple stain, negative stain and differential stain, which is further divided as an gram stain and acid fast stain. The detailed structure of larger microorganisms also cannot be effectively studied by the light microscopy, hence we discuss details of electron microscopy and its use. Third, the different types of media are used for the cultivation of bacteria, actinomycetes, fungi and viruses depending on their nutritional needs. Isolation and identification of colonies of specific microbes can be done depending on their morphological characteristics. Dear students, don't forget to review all of them in your self-study. If you have missed any of the module or content, you may log on to our website, that is www.cec.nic.in to download videos, FAQs, LOR and other contents so that you can stay updated with this chapter. Keep studying. Good luck. Goodbye.